Hello everyone, this is Sean Taylor, Field Application Scientist Manager for GenScript in North America. And as we head into the vaccination phase of the global SARS CoV-2 pandemic, tools will likely be needed to assess the level of protection that vaccines will offer between individuals and the timing required between vaccinations to assure ongoing protection globally. To this end, I'll be discussing an accurate and high-throughput measurement of neutralization antibody titers in serum samples from SARS CoV-2 infected, recovered, and vaccinated individuals. So, first, what are neutralizing antibodies? So, neutralizing antibodies are a special subtype of antibody in the immune response that block the virus from binding to the ACE2 receptor. So here we have a lipid bilayer membrane of a cell with, in purple, the ACE2 receptor jutting out, and the virus is docking to the ACE2 receptor via its spike protein, which dots the entire virus. In order to abrogate this binding event, we generate antibodies in our immune response, and some of those antibodies will bind in such a way to the virus, as we see here, that the virus can no longer bind to the ACE2 receptor. And these are called neutralizing antibodies. What we have done at GenScript, in conjunction with Dr. Lin-Fa Wang, was we created a kit that purifies the protein components of that binding event of the virus to the ACE2 receptor. The protein components that were purified include the receptor binding domain of the viral spike glycoprotein. That's right at the tip of the spike protein which dots the virus. So right at the very tip of the spike protein is the receptor binding domain that binds to here the ACE2 receptor. So we purified that protein, which is denoted here in brown. We've also purified the cytosolic domain of the ACE2 receptor, which is here in purple, denoted in green. So by coating plates, ELISA plates, with the ACE2 receptor, the cytosolic domain, and then presenting the purified receptor binding domain, which is conjugated to horseradish peroxidase, which helps to produce a signal in the assay, we're able to regenerate this binding event. So by incubating samples with the receptor binding domain conjugated to HRP, if those samples contain protective neutralizing antibodies, denoted here in red, then they'll bind to the receptor binding domain in such a way that the receptor binding domain can no longer bind to the ACE2 receptor, in effect neutralizing the ability of receptor binding domain to conjugate, to bind to the receptor binding domain and form that tight complex. When we wash the plate, all of these RBD HRP molecules will be washed away, and when we add TMB to generate a color which interacts with HRP, we'll get no color change. It will be clear. However, in the case where we don't have neutralizing antibodies, so only the blue non-neutralizing antibodies would be in this sample, which may bind to the receptor binding domain as we see here, but they don't bind in such a way as the red antibodies, the neutralizing ones, to block the ability of the receptor binding domain to bind to the ACE2 receptor and form a complex. So now all of these receptor binding domains are bound. And of course, because the HRP is bound to the receptor binding domain, which interacts and changes the TMB color to yellow, we get a yellow color. So if we don't have neutralizing antibodies, we get a color change. And if we do have neutralizing antibodies, we see no color change. And this is how the assay works. So in effect, we've taken away all of the viral components and are working with just the purified protein components that form this binding event to determine whether or not an individual has those special neutralizing antibodies that prevent the receptor binding domain from binding to the ACE2 receptor. So what are the advantages of this technology? The advantages include that it is immunoglobulin agnostic. So the receptor binding domain can therefore bind to any class of immunoglobulin, IgG, IgA, IgM, so long as that class of immunoglobulin generated neutralizing antibodies, which abrogate the binding of RBD to the ACE2 receptor. This is advantageous versus other traditional serology tests because now we have generated a total neutralizing antibody test. It doesn't matter which class of immunoglobulin those neutralizing antibodies come from, so long as they interact with the receptor binding domain, as we see here, then we will get no signal, which means that individual is positive for neutralizing antibodies. So that increases the sensitivity of this assay versus other serology tests that are specific for a particular class, IgG, IgM, or IgA, of antibodies. Also, the other advantage is that this assay is species independent. Again, because we're using purified protein components, it doesn't matter what animal the antibodies were generated in, so long as the animal was infected with the virus or the spike protein or receptor binding domain of the virus, they will likely generate antibodies to the receptor binding domain, and those antibodies will still block the ability of the receptor binding domain to bind to the ACE2 receptor. And again, this is advantageous for labs and companies that are working on drug development or vaccine development because they can use this test to follow the activity of their vaccines or drugs or antibody-based drugs from animal models to large animal models, primates, to humans. The other advantage of this assay is that the RBD is in solution. So in the kit, we provide the receptor binding domain conjugated to HRP in solution, which retains a more native conformation of the receptor binding domain and forces the neutralizing and non-neutralizing antibodies to bind very specifically to the native antigenic regions of that receptor binding domain, which increases the specificity of this assay versus other serology tests where those antigens, receptor binding domain or spike glycoprotein or nucleocapsid protein, are bound to the bottom of plates and their conformations can be altered and therefore non-specifically bind other types of antibodies that are unrelated to SARS-CoV-2, generating false positives. So we've seen in comparative studies a higher specificity of this assay as well. So not only do we get higher sensitivity from having soluble RBD as the bait protein, we also get higher specificity in this test. Of course, the third advantage of this test is that it's a dual detection and functional assay. So this assay, if it's positive, we know that the antibodies in the sample are neutralizing. They prevent the binding, they block or inhibit the binding of the receptor binding domain to the ACE2 receptor. So it's an added benefit that we're able to delineate the function of these antibodies. So there are a variety of tests that have been done with this kit, and this is one sample set of data from a company, Inatos Laboratories, where they tested 84 negative samples and 10 positive samples for SARS-CoV-2 with a number of different commercial assays that are traditional serology tests for IgG, IgA, IgM, and they assessed the specificity and sensitivity of the test versus the GenScript cPass test. And we can see that there was a pretty wide variation in sensitivity and specificity across the board. For these samples, the GenScript assay was perfect in both sensitivity and specificity. This translates to positive predictive value, negative predictive value, and accuracy, which are key measures of these types of ELISA assays for viral infections. And you can see again that there was a wide variation in the positive and negative predictive values and accuracy for all of these tests, these commercial serology tests, whereas the cPass test gave a perfect score for these particular samples. We also tested samples in conjunction with the University of Colorado and some other companies in the US, and determined by looking at 3 commercial kits using receptor binding domain coated plates, spike S1 coated plates, and nucleocapsid coated plates. We used 45 presumed positive samples and 23 pre-pandemic samples. And if we focus in on the pre-pandemic samples, which should be 100% negative, we can see that there was a variation in the number of false positives. So with the RBD coated plate, 3 of those samples were false positives. And for the nucleocapsid coated plate, there was 1 sample false positive. And we attribute this possibly to the fact that, again, in these plates, the bait protein is coated on the surface of the plate, may have altered conformation, and may be non-specifically binding other immunoglobulins. So we generate some false positives. The spike S1 coated plate was perfect in terms of the pre-pandemic samples, but for the positive samples, it gave a lot of false negatives. The RBD coated plate gave a couple, and the nucleocapsid coated plate gave a lot of false negatives as well. The cPass test recorded 6 false negatives, but interestingly, the 6 false negative samples out of the 45 that were tested were in common with both the nucleocapsid plate and the spike S1 plate. So it's uncertain whether these samples were simply just true negative samples or maybe did not seroconvert. We also did another study with 41 presumed positive and 27 pre-pandemic samples. And with the RBD and nucleocapsid coated plates and cPass, the negatives were all 100% negative. However, for the positive samples, again, the nucleocapsid coated plate gave a lot of false negatives. There was 1 false negative for the RBD coated plate and 1 false negative for the cPass coated plate. But again, that 1 false negative sample was in common with all 3 assays. So we are not certain that this was, in fact, a positive sample. It was likely a true negative sample. Taking both of these tests into account, adding up all the values, we determined the sensitivity, specificity, accuracy, positive and negative predictive values, and show that, again, as with the Inatos test, there was significant variation for these serology tests with coated plates, and the cPass test gave nice, very nice data in comparison to these tests for both specificity and sensitivity, and more importantly, for the accuracy, NPV, and PPV gave the highest results. We also compared the cPass test with live cell assays. So we wanted to show if the cPass test can correlate positively to the gold standard live cell tests that are being used to assess neutralizing antibodies. And in fact, with one study where we looked at 66 samples versus a live cell assay called plaque-reducing neutralization test, the correlation was very strong. The cPass test determined 40 samples as negative and 26 samples as positive for the 66. And for the PRNT test, at a high level of stringency, at a 75% plaque reduction or 90% plaque reduction, the results were almost the same, 39 negatives and 27 positives. So very close corroboration between cPass and the higher stringency PRNT test. For foci-reducing neutralization test, we looked at 45 presumed positive samples, and again, the results were quite close between cPass and FRNT at a 75% stringency, with the same 6 negative samples called negative for the live cell test versus the cPass test. So finally, let's turn our attention to a quantitative analysis. All of these data that I've shown you here are for qualitatively assessing the difference between positive and negative samples, and I hope you're convinced that the cPass test actually gives at least as good, if not better, quality and accuracy in calling samples between negatives and positives, with the added advantage of knowing that those samples contain neutralizing antibodies. But we also wanted to assess the ability of this test to work semi-quantitatively. And the workflow is described here. I'm not going to go into this in great detail. It's described in detail in the instructions with the kit, but suffice to say that the methodology is to dilute samples from using a standard curve. So we have a monoclonal neutralizing antibody that can be used as a standard with this assay that can be diluted. So we generate a standard curve from 150 nanograms per mil down to 2.34, measuring the OD values of that monoclonal neutralizing antibody, and then converting that to a percent inhibition. If we plot the if we plot the OD, the average OD value versus percent inhibition, so on this graph here, we can determine the OD at which we get a 30% inhibition. That's our cutoff from delineating between positive and negative samples. So we need to know where that cutoff is in terms of OD values. And then if we plot the standard curve by converting the concentration data on the log scale against the average OD 450 values on the y-axis, we can then plot a standard curve as we see here, very nice sigmoidal curve as expected, and note out where the 30% cutoff is at the respective OD value that we determined here. Then we can also determine the lower limit where we start to get the lower plateau of that sigmoidal curve, which is outside of the linear quantitative range. So really what we're trying to do is denote the linear quantitative range based on the upper limit, which is at the 30% cutoff, and a lower limit where the curves start to reach the lower plateau. And we'd like our titers for our actual samples to fall within this linear quantitative range. So then the next step in the process is to serially dilute the samples. So here's a low neutralization sample diluted from 1 in 10 to 1 in 2430. Same thing with the medium neutralization sample and a high neutralization sample. And we can see that for a low neutralization sample, we only need to dilute by 1 in 10 to be in this linear range that we have here for the individual samples. For a medium neutralization sample, somewhere between 1 in 30 and 1 in 270 is adequate to get a couple of points in this linear range. And then for a very high neutralization sample, we need to go all the way up to 2430 to get 1 point in the linear range. And then we can interpolate these data points we've chosen here onto the standard curve to get the titer values. By incorporating the titer values that we measured from the standard curve and multiplying by the dilution factors that we use, remember at the high neutralization level, we needed to dilute the samples by 2430 to reach the linear range. Then we get the final titers. So 23.4 times 2430 is this value, 18.8 times 90 is this value, and so on. And we determine fold differences between these samples. So this is how all this is done. This leads to data. So we looked at a number of samples and we were able to corroborate their neutralization values at a 1 in 10 dilution versus the titer values at the appropriate dilution to be within the linear range of the standard curve. And what we determined by looking at the difference from the lowest titer, which we denoted as 1, up to the highest titer, we just ratiometrically determined these values from this value here. So 237 divided by 220 is 1.1, and so on. We saw that for recovered infected patient samples, de-identified, there was a 410-fold difference in neutralizing antibody titers, which was quite astounding for us. We were not expecting to see such a large difference in the amount of neutralizing antibodies from one individual to another who had been infected and recovered from SARS CoV-2. So this lends the question and helps the question be answered surrounding what titer level is needed of neutralizing antibodies to assure ongoing protection from SARS-CoV-2 after vaccination or after infection. And we have a methodology now with this cPass test to actually measure this and compare this between individual samples. So just for regulatory approvals, this kit has been CE marked. It's been authorized by FDA under EUA for qualitative delineation between positive and negative samples. All the quantitative information that I've given you in the last slides have not yet been authorized by FDA, so our research use only. We have a number of solutions on our website for protein analysis and to generate kits. We have a lot of proteins available for SARS-CoV-2, including the antibody detection kits and ELISA kits that we talked about before. And we have services as well to help, if required, to generate assays in-house or purify large bulk quantities of proteins for work with SARS-CoV-2. So 1 out of 20 publications about COVID-19 have used genes or antibodies produced by GenScript, and this is how we say that we contributed to the fight against the virus.